Good day, everyone. With changing climate patterns, understanding crop resilience at a genetic level has become increasingly important. Today, we are presenting our study entitled Mathematical Modeling of Gene Regulatory Network for Flowering Time Regulation in Maize or maize under drought stress. Together with my co-researchers, Ms. Abutin, Ms. Gonzalez, and Ms. Pabines, and under the guidance of our advisor, Mr. Kenneth James Nugid, we will present the mathematical framework we've developed. Flowering time in maize is crucial for yield and survival. But what happens when drought strikes? Maize plays a vital role in food security, livestock production, and the livelihoods of million farmers. However, maize production is highly vulnerable to drought. During extreme events such as El Niño, yields can decline significantly, especially when drought occurs during the flowering stage. Flowering time is a critical phase in maize development. Even small changes in timing can greatly affect yield and plant survival. But flowering doesn't happen by chance. It is controlled by complex biological systems called gene regulatory networks, or GRNs. Although many flowering genes are already identified, we still lack quantitative evidence on how drought alters these networks and delays flowering. This leads to the main question of our study. How does drought reward gene regulatory networks to influence flowering time in maize? To answer this, we've developed a conceptual GRN based on the flowering pathways of Dong et al. 2012, then extended it by adding drought-related genes. In the leaf, the circadian clock pathway acts as the plant's internal timing system. The photoperiod pathway senses day length, where ZMCCT serve as the major flowering repressor and the main focus of our study. Lastly, the autonomous pathway includes ID1, which promotes flowering through internal developmental signals. In the shoot apical meristem, the aging pathway reflects plant maturity, while the GA pathway provides hormonal regulation of flowering. We then added drought tolerance genes such as ZMVOZ1, ZMARR16, ZMMPK3, and ZMHY5. Under drought stress, these genes influence stress response signaling and indirectly regulate ZMCCT, allowing the plant to adjust flowering time for survival. Since modeling every interaction would make the system too complex, we focus only on genes with strong biological evidence and major roles in flowering regulation and drought response. This simplification allows us to focus on ZMCCT as the main integrator of the network while still capturing the essential mechanisms behind flowering regulation and drought response. To mathematically model the GRN, we use Hill functions to represent gene interactions. Activating Hill functions describe genes that promote expression, while inhibiting Hill functions describe genes that suppress expression. These functions are defined by parameters such as synthesis, degradation, threshold, and cooperativity. We then formulated the system using ODEs. Each equations describe how gene expression changes over time through synthesis and degradation processes. Table 1 presents the notation used in the simplified GRN, where each gene is represented by a variable, say sub I, corresponding to its expression level. Finally, we obtain a system of 13 ODEs describing the dynamics of the genes in the simplified network. Equations 1 to 13 were directly constructed from the GRN structure.
We now proceed to the analysis of the proposed model to ensure that the model is mathematically well-defined and the results are biologically meaningful. For existence and uniqueness, we use Cauchy-Lipschitz theorem. Since the Hill functions and linear degradation terms are continuous and satisfies the Lipschitz condition, the model admits a unique solution for any given initial condition. The model was also shown to preserve positivity and boundedness. Assuming non-negative initial conditions, all gene expression levels remain non-negative and bounded over time, ensuring biologically feasible behavior of the system. Then, boundedness is established by considering the total gene concentration, ensuring that expression levels stay within realistic ranges. Overall, the model is well posed, with solutions that remain unique, positive, and bounded over time. The Jacobian matrix evaluated at the equilibrium point characterizes the local behavior of the system, while its eigenvalues determine the local stability of the equilibrium. Since all computed eigenvalues are negative real numbers, it indicates that the equilibrium is locally asymptotically stable. Biologically, this suggests that the flowering time gene regulatory network remains robust and stable despite small perturbations, supporting reliable flowering behavior even under drought stress. For parameter estimation, we use time series gene expression data from Arabidopsis thaliana. a well-established model organism. Although the study focuses on maize, this is justified due to conserved gene regulatory behavior across plant species. We applied here the Generalized Profiling Method, or GPM, an established framework for parameter estimation in differential equation models. This procedure involves an inner optimization for spline fitting and an outer optimization for estimating the ODE parameters. This ensures consistency between the model dynamics and the observed data. Using GPM, we estimated synthesis and degradation rates for both normal and drought conditions. We also obtained threshold and Hill cooperativity parameters governing gene activation and inhibition. These parameters define the regulatory interactions among genes in the simplified network. Furthermore, we performed quantitative validation using symmetric error integral to compare the simplified and conceptual networks. The results show low error values under both conditions, indicating that the simplified GRN preserves the essential dynamics of the original system. Next, a sensitivity analysis was performed using Latin hypercube sampling and partial rank correlation coefficients. The results revealed that parameters associated with ZMCCT, synthesis, and degradation have the strongest influence on flowering time dynamics under drought stress. To explore gene expression dynamics, we perform simulations under two conditions: normal or well-watered, and under drought stress. 3 simulations are conducted to compare the behavior of the system under these conditions. For the first simulation, it indicates that under normal conditions, gene expression converges to stable profiles, while drought conditions led to altered expression dynamics, reflecting the impact of environmental stress on network regulation. Simulation C highlights the effect of drought stress on ZMCCT expression. Under normal conditions, ZMCCT reaches a higher, more sustained level, while drought stress suppresses its long-term activity. However, ZMCCT shows a rapid initial increase, indicating an early stress response before gradually declining. The simulation also reveals the expression dynamics of ZMM4, a key flowering marker. Under normal conditions, ZMM4 rises quickly, indicating an early flowering initiation. Under drought stress, ZMM4 increases more slowly at first, but eventually reaches a higher steady level, indicating a delay in flowering onset, but sustained signaling once flowering begins. The next simulation shows the behavior of the GRN under drought stress when the synthesis level of ZMCCT is varied, while keeping all other parameters unchanged. Under drought conditions, the simulations show that reduced and knockout ZMCCT lead to elevated expression levels in most downstream flowering-related genes. In contrast, increased and overexpressed ZMCCT are associated with generally lower expression levels across most genes. The third simulation focuses on the expression dynamics of ZMM4, where the flowering time is defined by the timing of its peak expression. Under normal conditions, ZMM4 expression increases rapidly and shows only minor differences in peak timing across ZMCCT regulatory states. Under drought stress, ZMM4 rises more slowly and peaks later overall. Relative to the drought baseline, the knockout, increased, and overexpressed ZMCCT exhibit earlier ZMM4 peak timing, whereas the reduced ZMCCT shows a delayed peak.
This study, we addressed the major research objectives by developing and analyzing a continuous gene regulatory network model for maize flowering time regulation under drought stress. The model was shown to be mathematically well-posed, and its behavior was examined through stability analysis, sensitivity analysis, and numerical simulations under normal and drought conditions. Most importantly, this study demonstrates that drought consistently delays flowering relative to normal growth conditions. Under drought stress, complete knockout of ZMCCT results in substantially earlier flowering relative to the drought baseline, while partial reduction of ZMCCT leads to moderate delay. In contrast, increased and overexpressed ZMCCT also produce earlier flowering relative to the drought baseline. Together, these results indicate that the effect of ZMCCT on flowering time under drought conditions is not linear. Importantly, these findings do not suggest that elevated ZMCCT promotes flowering biologically. Rather, they reflect non-monotonic regulatory dynamics within the modeled network, in which extreme perturbations of ZMCCT alter the timing of downstream floral commitment signals. Overall, this work shows that SAM-CCT functions to modulate the severity of drought induced flowering delay through complex network interactions, rather than acting as a simple dose-dependent regulator. And so that concludes the presentation of our paper. Thank you for your valued time, interest, and continued guidance.